I now uh, hand over the today's agenda to the Director CSA and East. It will be started by felicitation to the Director from Eco Concepts.
Very good morning. Dr. R.C. Borwan and all my senior colleagues, Mr. Das, and all the dignitaries, invitees, participants, NGOs, and ladies and gentlemen. This is again a unique program, a workshop, one-day workshop, which is being organized. And of course, the initiator is Mr. Das, because he is the one who has approached us that this is an important issue, that whether we can be a part of it. And without any hesitation, without thinking even twice, how we are going to do, what we are going to do, but we have accepted it because this is a very important issue that concerns each and every one of us, and also concern to the society in general. So on this occasion, on behalf of all my colleagues, on behalf of each and every one, I have the great pleasure of welcoming each and every one of you to our institute, and hope and wish that a deliberation such as this, or the discussions that we have in such a meetings such as this, which is again very specialized, would go a long way. When it comes to the pollution, of course we have all types of pollutions. Air pollution, water pollution, soil pollution, whatnot, every pollution, and of course it is also outcome of industrialization to some extent, but negligence also to a greater extent, that we don't really take certain things as serious as deserve to be. So in this context, this awareness is also very important. Identification of the problems is also important, and also not only the awareness, identification, but then finding the solutions is much more important. How do we solve these solutions in a sustainable? That is most important. So on this occasion, on behalf of all my colleagues, we congratulate these NGOs for volunteering, for taking the part in such novel initiatives which concern the society, and Mr. Das in particular, and also the Usha, also who has been also I think actively involved, and each and every member. But I can see that I think I was told that all the people who are involved here are mostly contributing through almost like a voluntary. I think we, on behalf of all my colleagues, I think let us give a big hand to this, a team of people who are working for society. So with this, now I, with welcoming remarks, and now I request Mr. Yan Das to briefly enlighten us the purpose of such a meeting and the objectives and other details.
A very good morning, one and all. Good morning to you, sir, and good morning to all the scientists present here, and the NGO people, government dignitaries, and everyone. So, as sir has mentioned that I need to highlight the purpose of the discussion. Well, let me first and foremost tell you that there is a network of individuals, organizations, groups who are working on the issues of water quality and arsenic in general. And this network started around 2.5 years back. And the network headquarters is based in Secunderabad. That's what you get in the kit that you get, Saki Waters. That's the headquarters, but that's irrelevant. The network is a group of all like-minded people like us who brings in knowledge, then ideas, and all those kind of people who are working in the sector. And the network came to Assam. And it was in the month of September that there was one discussion in IIT Jorhat, where right from government people to community people to scientists, research scholars, everyone participated. And to find out ways actually what can be done for the solution of this problem. You know, enough has been said about this problem, enough of research, so much of research is coming out day by day. And Sir Unni was also saying, telling me that once you have the knowledge built, so what on ground? So this is the primary question, what on ground and how on ground? So precisely this is the purpose of this discussion. Now after this discussion, a group of delegates went to Nerivalam and they met Mr. Manoranjan, and this idea of having several discussions in different districts of Assam was actually coined by him, and we are actually following it up. So for the network to coordinate its activities, to have all of us in the group, we at EcoConcept are trying that up. But other than that, it's all of us here today. All of us here. So much of knowledge. I'm really excited that so much of knowledge, so much of ideas will pour in today. And it's all up to us at the end of this day, how do we carry these ideas and knowledge into practice. So that's it for the purpose. It's very simple, that once the knowledge pour in, so how do we take it back to the community? What are the ways we can take it back to the community? That's it. Thank you, sir.
Yeah, thank you, Mr. Das, for giving us a brief introduction to the purpose of organizing such a workshop. Now, the next item in the agenda is a self-introduction by all the delegates, all the participants who are present here. So maybe you please, I would request them to, so where they come from, what they have been doing, and what, just to give their, biodata, basically to enlighten all of us, and how they are excited about it, in working on such a project. Okay, thank you. So we start from where?
Hello, good morning to you all. Myself Dr. Prabhat Kotuki. I am working in the analytical chemistry department. Basically, I am a geochemist. I am working a lot on this water pollution and water problems of this northeastern region. And with this background, I am here today to share the knowledge and to have interaction with the people, those who are working on the water quality parameters. Thank you.
Dr. B.G. Unni. Basically, I am a biochemist working at the biotechnology department as the chief scientist and also the area coordinator of biological sciences. In fact, this is a very serious problem. That's why I cut off, in fact, 2 major conferences in between came here. For the last 10 years, we had a network projects related to environmental, taken out the paper mills, and then oil refining and the coal mines areas and all. Accidentally, we found quite, a very high concentration of arsenic near to the rural areas when we analyze, because we go with the soil, water, food, air. So what I found for the last, I got the information about you have a discussion at Guwahati and as well as at the Tezpur. See, a time has come now, we should stop giving more details on the research, whether high pH will do arsenite more, low pH will do arsenite less. We should come up with a very conclusive evidence that how to control. There are filters are available. What is happening with the filter? They accumulate the entire arsenic concentration from a low toxicity to high toxicity, then where you will dump the filter. That means the place where you are dumping the filter will more pollute. So we did some sort of a work. It's a very preliminary study using as a biomaterials. Maybe a plants, I'll talk when I'm going to present there. There, this thing has got a detoxification mechanism. If this metals, toxic metal will be used as a cofactor, as a coenzyme. They are like water hyacinth. Water hyacinth is a very good plant material to detoxify mercury, because the mercury used as one of the cofactors. Same thing like arsenate also we have done with some plants, where it detoxifies to arsenite and then a complex form and is stored in the cytoplasm. So some sort of studies we have done with the research, plus it can also be used as a biomaterial. So this will be presented. That is the area we have done it, and let us see how far it is going to be utilized for your ecosystem. Thank you.
Thank you.
Good morning. I am Jorhat, Chief Scientist, Geoscience Division.
Good morning, everybody. I am Pranav Bhattagoti. I am heading the Chemical Engineering Division of this institute. We are also working in this water purification system. Recently, we have completed one project with Material Science Division collaborating with them. Dr. R.L. Goswami is deeply involved, and our scientists also involved there. So they have been working on mitigation of fluoride from drinking water. So they have developed a process, and based on that, got another project now for subscale for development. And we'll be pleased to provide engineering input for development of some equipment wherein the fluoride can be removed from the drinking water and can be used in a low-cost way. And maybe that will be useful for this sort of arsenic mitigation also. Thank you.
Thank you.
Good morning to all. মোৰ নাম পিনাকি সেনগুপ্ত, মই ইয়াত মেটেৰিয়েল চায়েন্স এৰিয়া কোঅৰ্ডিনেটৰ। এই এৰিয়াত ডক্টৰ R.L. গোস্বামী যিজন আমাৰ চিনিয়ৰ চাইণ্টিষ্ট এজন কোঅৰ্ডিনেট কৰি আছে। তেখেতে আমাৰ এই এৰিয়াত আৰ্ছেনিক আৰু ফ্লৰাইড ৰিমুভেলৰ তেওঁলোকে যথেষ্ট কাম কৰিছে আমাৰ কেমিকেল ইঞ্জিনিয়াৰিং ডক্টৰ R.L. গোস্বামীৰ লগত। বৰ্তমানে তেওঁ এটা DSTৰ প্ৰজেক্ট এটা আৰম্ভ কৰিছে, য'ত তেওঁ এটা ফ্লৰাইড ৰিমুভেলৰ তেওঁলোকে এটা ডেমনষ্ট্ৰেশ্যন প্লান্ট কৰি কিছু গাঁৱত সেইটো ইমপ্লিমেণ্ট কৰাৰ চেষ্টা কৰিম। ইয়াৰ বাহিৰে মেটেৰিয়েল চায়েন্সৰ ভিতৰত কোল কেমিষ্ট্ৰি ডিভিজন আছে, য'ত কোল কেমিষ্ট্ৰি ডিভিজন কোল মাইনিং এৰিয়াত যিটো এটা ডাঙৰ এনভাইৰনমেণ্টেল প্ৰব্লেম, সেইটো হৈছে এচিড মাইন ড্ৰেনেজ বুলি কয়, যি গৈ পেলাই খোৱা পানীত কণ্টামিনেট কৰি আছে। তেওঁলোকে তাত বহুতো ভাল কাম কৰিছে আৰু তেওঁলোকে তাৰ মেথডছ উলিয়াইছে কেনেকৈ প্লান্ট স্কেলত, কিন্তু আমি ৱেইট কৰি আছোঁ আমি কেনেকৈ তাক ফিল্ডত ইমপ্লিমেণ্ট কৰিব পাৰোঁ। তো মই মিষ্টাৰ দাস আৰু আমাৰ এইফালে ডক্টৰ গোস্বামী আছে, তেওঁলোকক ৰিকুৱেষ্ট কৰিম যে মই এজেণ্ডাত দেখিছোঁ কিছুমান ঠাই আছে তেওঁলোকে স্কুইজ কৰিব পাৰিব। যদি স্কুইজ কৰিব পাৰে তেওঁলোকে আমাৰ এই যিবিলাক বিশেষকৈ বাহিৰৰ ডেলিগেটছ আছে তেওঁলোকক কেইটামান পাৰ্টিকুলাৰ ডিভিজন যদি ইয়াত খুব কম সময়ৰ কাৰণেও হয় যদি ভিজিট কৰাব পাৰে, that can be a very nice thing for them, experience জানিব পাৰে।
I'm Alokananda Sengupta and I am leading the Planning and Project Monitoring Division, and I am happy to be associated in the monitoring of this project. Thank you.
Good morning to you all. I am Dr. Shashi Borwa, heading the Petroleum and Natural Gas Division of this institute.
Good morning, everyone. Myself Saurabh Das. I have come from Assam Agriculture University. I am a PhD thesis scholar working on trying to isolate some arsenic tolerant bacteria and trying to see whether they help in the mobilization of arsenic actually through their respiration process actually. Thank you.
Thank you.
Good morning, everyone. Myself Kuldeep Talutra. I have come from Assam Agriculture University, presently doing my PhD in Department of Plant Pathology, and I am doing my research on arsenic, and thereby we are trying to find some arsenic mobilizing bacteria, and whereby we are going to find some biodetoxifying mechanism through which we can decontaminate the arsenic in the soil. Thank you.
মই বিমান শইকীয়া তিতাবৰৰ পৰা আহিছোঁ। মই ষ্টান একাডেমি চাইল্ড অৰ্গেনাইজেশ্যনৰ চেয়াৰমেন আৰু তিতাবৰ চুঙিৰ অৰুণোদয় হাইস্কুলৰ হেডমাষ্টাৰ। থেংক ইউ।
মই অনন্ত খনিকৰ তিতাবৰ পৰা আহিছোঁ। মই ৰাইজৰ মাজত পানীৰ পৰা হোৱা যিখিনি অসুবিধা বা বিভিন্ন ধৰণৰ অসুখৰ আমি যিখিনি প্ৰত্যক্ষ কৰিছোঁ সেইখিনি নাইকিয়া কৰিবৰ কাৰণে আমি কিছুমান প্ৰচেষ্টা লৈ আছোঁ আৰু মই মেলামাত্ৰী গভ জুনিয়ৰ বেচিক স্কুলৰ হেডমাষ্টাৰ।
Good morning to all of you. মই অনন্ত কুমাৰ শৰ্মা। মই এই অনুষ্ঠানৰে এজন ৰিটায়াৰ্ড প্ৰিন্সিপাল টেকনিকেল অফিচাৰ আছিলোঁ। মই এই অনুষ্ঠানত যোৱা 36টা বছৰে মই পানীৰ যিটো সমস্যা সেই সমস্যাৰ লগত ওতঃপ্ৰোতভাৱে জড়িত আৰু উত্তৰ পূৰ্বাঞ্চলৰ বিভিন্ন অঞ্চলত আমাৰ এই অনুষ্ঠানৰ পৰা অৰ্গেনাইজ কৰা বিভিন্ন ধৰণৰ যিটো এৱেৰনেছ প্ৰগ্ৰেমবিলাকত অংশগ্ৰহণ কৰি পানীৰ পৰা হ'ব পৰা বিভিন্ন ধৰণৰ অসুবিধা ৰোগ আদিৰ বিষয়ে মানুহক সজাগ কৰিবৰ কাৰণে আমি চেষ্টা কৰিছিলোঁ আৰু আপোনালোকৰ হাতত বুকৰ এখন ৱাটাৰ ইজ লাইফ নামৰ কিতাপ এখন খুলি দিয়া হৈছে সকলোৰে হাতত। এই কিতাপখনৰ সংকলন মই আৰু ডক্টৰ বিনোদ গগৈ কৰিছিলোঁ আৰু আমি এই উত্তৰ পূৰ্বাঞ্চলৰ বিশেষকৈ পিছপৰা অঞ্চলত আমি অৰুণাচল নাগালেণ্ডকে ধৰি আৰু অসমৰ যিসকল পিছপৰা অঞ্চল সেই অঞ্চলত বিভিন্ন ধৰণৰ আমি এই সজাগতা অনুষ্ঠানবিলাক কৰি পেলাই মানুহৰ মাজত মানুহক সজাগ কৰিবৰ কাৰণে চেষ্টা কৰিছিলোঁ। ধন্যবাদ।
Good morning, everyone. Myself, Surupak Kumar Gogoi from PACD Jorhat. I am working as a DWSF consultant, ICSRD.
Good morning to everybody. I am Mr. Provin Kumar Hindi. Basically, I am a civil engineer coming from PACD department. As a public engineer, our responsibility is to provide potable water, basically arsenic-free water to the community. Various steps have been taken from our department to provide potable water in the arsenic zone area. As a personal effort, I am writing various types of articles, particularly arsenic contamination in groundwater in local newspaper to aware and sensitize the community. Thank you.
Thank you.
A very good morning to all of you. Myself, Dr. Medul Setia. I am coming from Jaigaon, Gulaga district. I did my PhD in this particular subject, arsenic and its mitigation in Gulaga district actually. So today I'll be very happy if I can able to share my knowledge and thoughts to all of you. So thank you, sir.
Good morning. Myself, Dr. Navajoti Soikia. I am coming from Kaziranga University. I teach chemistry as an associate professor and head of the department.
মই আৰাধনা গোস্বামী কৈছোঁ। মই ডক্টৰ পিনাকী সেনগুপ্তাই ক'লে মই পানীৰ ওপৰত কাম কৰি আছোঁ। আমি এটা লোকেলি বায়'মেটেৰিয়েলৰ পৰা এডজাৰ্বেণ্ট বনাই পানীৰ ফ্ল'ৰাইড ৰিমুভেলৰ এটা প্ৰজেক্ট শেষ কৰিলোঁ আৰু এটা প্ৰজেক্ট আমাৰ DSTত এপ্ৰুভ হৈ গ'ল। সেইটো অকণ স্কেল আপ কাম আমি কৰিবলৈ ধৰিছোঁ আৰু মই ফুড আৰু নিউট্ৰাচিউটিকেলৰ লগত কাম কৰিছোঁ। সেইখিনি আমি ধান আৰু ধানৰ ৱেষ্টৰ পৰা ৰাইচ ব্ৰেনৰ পৰা আমি নিউট্ৰাচিউটিকেলৰ কাম কৰিছোঁ।
Good morning, everyone. My name is Usha Devani and I work for India Water Portal. It's a web-based platform on water and related issues in India and I cover the northeast region for the portal.
Good morning to everyone. Myself Monuranjan Man. I am coming from Inaugural, Northeastern Regional Institute of Water and Land Management. Last 14 years, মই পানীৰ ওপৰত কাম কৰি আছিলোঁ। 14 বছৰ ধৰি আৰু আমাৰ অনুষ্ঠানতে প্ৰথম অসম আৰু উত্তৰ পূৰ্বাঞ্চলৰ পানীত আৰ্ছেনিক থকাটো আমি গম পাইছিলোঁ আৰু আমি এটা প্ৰজেক্ট কৰিছিলোঁ এইটোৰ ওপৰত। তেতিয়াৰ পৰা আমি আৰ্ছেনিক আৰু ওপৰত কাম কৰি আছোঁ কিছু পৰিমাণে। বৰ্তমান আমি আৰ্ছেনিক মিটিগেশ্যনৰ ওপৰত কিছু কাম কৰি আছোঁ তেজপুৰ ইউনিভাৰ্চিটিৰ কেমিকেল চায়েন্স ডিপাৰ্টমেণ্টৰ লগত জড়িত। তাৰোপৰি বৰ্তমান আমি যাতে এই সমস্যাটো ৰাইজৰ মাজত কিছু শিক্ষা দিব পাৰি তাৰ কাৰণে আমি NGO আৰু কিছু পাব্লিক হেল্থৰ মানুহক আমি ট্ৰেইনিং দিওঁ। ঠিক তেনেকুৱা এটা ট্ৰেইনিঙতে আচলতে মই দাসক লগ পাইছোঁ, মোৰ ট্ৰেইনিং কৰিব আহিছোঁ। I organized one training, sir, and he was one of participants of this training, and glad to hear our sir, that immediately I'm try to be part of this organization. So really we feel that we want to bridge from science to the public, and NGO is the good thing for bridging them, because scientific staff have not so much time to go to the people. So on the ground, I told them that you will be the right person to work on that. That's why I try to organize at least 4 to 5 trainings, early 3 days or 5 days training on water quality monitoring for NGO specific. And this is my really when they come to me and they are friendly organization, then I told them that a very big if you organize a regional based conference on that, then only a few person will get comment like that. Only 2 or 3 scientists may be join to get a workshop if you organize a Jorhat. But if you organize on district level, small workshop, 1-2 workshop, then we can make a ground level worker who are working in the ground. So we can know that what is the ground problem. Then only we can think for a bigger workshop so that we can take some decision or take some come up with some solution that what we should praise the government or like that. So before that ground work is
very essential.
And that's why I think they come with that small workshop. Like this, they are going on Facebook and some states. I told them that some
Thank you. Yeah, every positive step adds to the reaching the goals.
So I have the mic for the second time. Okay, so I'm Inaugural. I'm from the organization Eco Concept. So we primarily work on environment education with the children, especially focused in the rural areas, and we work in Kamrup district. So I forgot to mention one important point, that the purpose of this workshop doesn't end here. So after this workshop, there are a series of activities where all of you in your individual capacity or organization capacity can contribute. So the next activity that is thought of as of now is a health camp in the Titabor area, and followed by an epidemiological study in the area in order to understand the depth of the problem and in order to understand how the community is responding to the problem. So maybe all of us in our whatever capacity, we can be a part of that process.
Okay, thank you very much.
Good morning to everybody. I am Dr. R.C. Burua. I am the area coordinator of Chemical Sciences and Management Sciences. Thank you.
Good morning to all. Myself Amarjit Rai, STD in the Public Health Department.
Namaskar. Good morning. মোৰ নাম দিলীপ কাকতি, মই পাব্লিক হেল্থ ইঞ্জিনিয়াৰিঙৰ এছিষ্টেণ্ট এক্সিকিউটিভ ইঞ্জিনিয়াৰ। মই তিতাবৰ চাব ডিভিজনৰ আজি 5 মাহে কাম কৰিবলৈ মোৰ সৌভাগ্য হৈছে। তিতাবৰ আপোনালোকে জানে যে তিতাবৰৰ 17টা গাঁও পঞ্চায়ততে আৰ্ছেনিক পোৱা গৈছিল। তাৰ কাৰণে প্ৰায় 100 কোটি টকা খৰচ কৰি গভৰ্ণমেণ্টে গ্ৰেটাৰ তিতাবৰ ৱাটাৰ ছাপ্লাই স্কিম ইমপ্লিমেণ্ট কৰিছে। সেই স্কিমটোৰ চোৱা চিতাৰ দায়িত্ব মই 5 মাহে কৰি মোৰ যিখিনি এক্সপিৰিয়েঞ্চ হৈছে সেইখিনি আজি মই অলপমান শ্বেয়াৰ কৰিম। আৰু দেখিছোঁ বিভিন্ন ছেক্টৰৰ পৰা মানুহ আহিছে, আমি বহুত কিছু কথা আজি জানিম। ধন্যবাদ।
Morning everybody. I'm Dr. Brandon Tamoli. I'm a physician by profession and I'm a first year postgraduate student in Jorhat Medical College in the Department of Community Medicine.
Thank you. Good morning friends. I'm Professor Vishnuram Das. I'm currently heading the Community Medicine Department in Jorhat Medical College. So my purpose of coming here, because from the many sectors, people like you who have already done many research in the field of arsenic and all, how this research work or research finding can be translated at the community level, or what is the direct benefit of the research to the community people.
So I am very agree with Dr. Nirupal Das. He has rightly mentioned that epidemiology of the arsenic problem in the block of Titabo. How we can formulate the epidemiological studies, and after that, how we can plan and provide an implementation package to the community people directly. So that's all. Thank you very much.
Good morning to everybody. I am Dr. Devasis Panda, registered Kaziranga University. I did my PhD actually on environmental geochemistry, but I was not doing research for last couple of years. Now I am interested to do some research about in this area. That's why I come here. Thank you.
Thank you. মই তিতাবৰ পৰা আহিছোঁ। মোৰ নাম মহম্মদ তফিজুল আলী, মেলামাটি জুনিয়ৰ বেচিক স্কুলৰ SMC প্ৰেচিডেণ্ট।
Good morning everyone. Myself Miss Pompi Bora. I am coming from Tittabur and I did MA in Political Science from Tibor University. Thank you.
I am Jorhat Bora, works in General Engineering Division of NISA. Basically, I am a mechanical engineer, presently involved in different projects where engineering inputs is required. Thank you.
Hello, good morning everybody. Myself Tomil Hussain Ahmed, working in the chemical engineering division. Basically, I am a chemical engineer, and as you all know, we are doing some projects on defluoridization of drinking water, and for that particular project, my involvement will be to design basic engineering equipment like backplate column and start pilot plant study. Thank you.
Good morning everybody, and thanks to all of you for participating in this noble venture. I am H. Harambo Prasad Sharma. I am a retired scientist and was heading the chemical engineering department of this institute. During my colleagues were working and have been working in water pollution in various aspects. And particularly I can mention one industry which has implemented our design for water pollution that was contaminated in oil-free water. That was contaminated with a lot of oils and other contaminants, metals, etc. So we devised certain methods using chemical as well as biochemical materials to mitigate water pollution. As you see, in the northeastern region, not only arsenic and fluoride is the contaminants, main contaminants may be, say, iron also, and oil, all these things are here. So we have to look more into those aspects also and see how we can translate our findings into our community development with easy and economically affordable methods, so that there will not be further contamination or downstream contamination. So we have to see introduce this. Thank you.
Thank you.
সমবেত সকলোকে শ্ৰদ্ধা জনাইছোঁ। মই আহিছোঁ সংকল্প শিক্ষা নামৰ এটা বেচৰকাৰী সংগঠনৰ ফালৰ পৰা। আমি ইতিমধ্যে ধঁপাতমুক্ত অঞ্চল গঢ়িবৰ অৰ্থে আমি কিছু কাম কৰি আছোঁ। ধঁপাতৰ অপকাৰিতা সম্পৰ্কতে এৱাৰনেছ কেম্প আমি কৰিছোঁ, স্বাস্থ্য সজাগতা সম্পৰ্কত কৰিছোঁ, আৰু ইতিমধ্যে আমি এই প্ৰতিষ্ঠানৰ ৰাজীৱ গোস্বামী ছাৰক লগ পাই আমি আৰ্ছেনিক পানী সম্পৰ্কীয় কৰ্মৰাজি কৰিবলৈ এটা উৎসাহিত হৈছোঁ। সেই উদ্দেশ্যে আজি আমি ইয়াত সমবেত হৈছোঁ। মই আশা কৰিছোঁ ইয়াত সমবেত হৈ বিভিন্ন জ্ঞানগ্ৰস্ত ব্যক্তিসকলৰ যোগেদি বহুখিনি কথা জানিব পাৰিম। মোৰ নাম জগন্নাথ অগ্নি বৰা।
মোৰ নাম বিদ্যুৎ শইকীয়া, মই যোৰহাট বাহনৰ পৰা আহিছোঁ। মই সংকল্প শিক্ষা নামৰ এটা বেচৰকাৰী অনুষ্ঠানৰ এজন সদস্য। মোৰ পেছাত মই গোৱালপাৰা ডিষ্ট্ৰিক্ট জাজৰ এজন ষ্টেনগ্ৰাফাৰ হয়।
Good morning everyone. I am Dr. B.K. Medhi from Assam Agricultural University in the rank of Associate Professor.
So as far as the block of Titapur is concerned regarding arsenic contamination, already I did some work. GPS-based arsenic vulnerability zone already whole work has been completed. So if you provide us the GPS point, each and every point of the Titapur block, I can immediately do the exact arsenic contaminated zone, say, in respect of groundwater and soil. So regarding fluoride also I did some work on Margarita subdivision. So if given the opportunity, I will show some work already. Recently I have submitted one project in our atomic research station. So already that project has been approved. Because this arsenic contamination, always we are talking about arsenic contamination, groundwater arsenic contamination regarding drinking water and all aspects. But as you all know that Assam is the monocrop area, rice-based area. So arsenic through rice, there is already arsenic entered into the food chain. So this is the most dangerous aspect. So we will discuss. Thank you.
Thank you very much. Good morning, everybody. I am R.L. Goswami. I am organizing on behalf of Director R.L. Jorhat, the local organizing committee of this workshop. My interest in water is in the field of fluoride and arsenic is there. Apart from that fluoride and arsenic, I have also worked on environmental gas pollutions like toxic sulfur dioxide and hydrogen sulfide using nano oxides.
Good morning to all of you. Myself, Ingi Soitya. I am a project fellow in Material Science Division in this institute under the guidance of Dr. Rajiv Goswami, and my project work is nano-oxiding membrane reactors by chemical approach. Thank you.
Good morning. Myself is Shweta Sharma, and I am a project fellow in Material Science Division. Good morning. Myself, Gayatri Global. I am working as a project fellow in Chemical Engineering Division.
Thank you. Yeah, thank you all for introducing very briefly and also enlightening us your various activities. That is very nice. The next item is speech by the director. I don't think I have to really give a speech because we are running shortage of time now. I think I'm sure you must have scheduled several presentations at various groups. Because your program also doesn't give the names.
Actually, we had a lot of presentations and we had to sort out some 5 presentations. Like it was like not very sure till the last moment.
So before I just request my colleague Dr. Asim Borba to say a few words, I would like to just tell you because I see there are several new guests to our campus. And first, I must apologize because I can't speak in Assamese. Please excuse me for that. But let me tell you very briefly about our institute. So our institute is one of the oldest institutes. It was started almost way back in, established in 1961. And since then it has been, working on various activities, not only with the society but also high-end research activities. And all our activities can be broadly classified into major areas of chemical sciences, biotechnologies, biological, agrotechnologies, engineering sciences, geological sciences, material sciences, and you name it, an activity, we have it here. Therefore, ours is a truly interdisciplinary research laboratory. And, we have also developed several technologies. Again, these are cottage-based, starting from the low dust chalk to the liquid deodorants, to the nutraceuticals, to the cement plants, and then biopesticides, and also several plant-based affordable healthcare products like anti-arthritis, antifungal, because some of these are already in the market. And we are indeed very happy to share with you that some of these projects are really bringing a lot of visibility to our institute because these were found to be quite effective and efficient. And our institute is one of those, actually, one of the best performing institutes with respect to the activities related to the CSR agenda programs. That is mostly the working with the society and for the society benefits, like cultivation of the mushrooms, cultivation of the lemongrass, cultivation of the citronella, and establishing various distillation plants, which have really helped the society to a great extent. But there is also scope for us to reach to the society in various other forms, and that is where when Mr. Das has approached us that this is a very important problem to this region, particularly the water contamination with the fluoride and arsenic. And our institute also started way back working in these activities, particularly we have developed some water filters, candles, and all that, but that was stopped somewhere in between. But then recently again we have started working on particularly the purification of water, at least free from fluorides and arsenic contents. And this is still taking off, and it has to be demonstrated at the village levels. And I think the next phase of this activity is to try it out in the villages and see how we can help the society. So that is broadly about our institute, because we have almost like 110 approved strength of the scientists, and then we have a huge helping hands like technical officers, technical assistants. Total we have almost like 450 permanent strength. But again our backbone is the students, and the number is going up every day. And I think we have almost like around now 250 to 300 students with us under various programs as a project assistants as well as research students working in various divisions of our institute. So that is briefly about our institute. So since this is very important to the society and pollution, it is anything related to the water or energy or environment, it is very important. And it is going to go in a long way, because somehow for various reasons for several years, particularly in India, we have not given importance to these activities. And still we take that many things for granted. Industries also take them for granted, and their main aim is to just make profits at whatever the cost. But fortunately, things are changing. Government policies are changing. Government is introducing several regulations, and it is for the good. And industries also have realized that they have to play a major role, that they cannot really contaminate the air, environment, and water no longer. That unfortunately they used to do. Many of the industries used to do in earlier cases. But now stringent regulations are going to be there, and I think much more stringent regulations are going to come into operation, and everyone has to play. Not only the industries have to play, not only the government has to play. Each and every individual, from the the organizations and NGOs, and each and every individual has to play a role in this. Because again, in this context, I think the Prime Minister's the recent slogan is a Swachh Bharat is one of these. Because if you can remove the dirt and filth and from the streets of India, of course certainly it is going to add in a big way to to our country and help the society. So these are all have to be implemented, but Prime Minister alone cannot implement, or the government alone cannot implement. It is we have to get involved. It is we have to take it positively. It is we have to feel that it is very important. We have to own it and make it success. Any program that we take, even water purification, it is very important. Health is important. I think there is a saying that health is wealth, and it is, I would say, it's much more than wealth, because wealth, if you lose it, you can get it. You can change the position, you can go to the by R&D to the, you can go to the industry, work and then make money. So I think health is much more than wealth, because but if you lose health, you will lose something, and but of course if you lose character, you lose everything. That is a different story. But therefore, it is very important. It is concerns all of us, and what we drink, what we eat, what we breathe. It is very important, and in that context, this is in a small way. It's a big way. I congratulate not only Das and his team and also all the NGOs, but each and every one who are part of this program, and I wish this initiative a grand success. Thank you.
Answer and discussion time. So the first presentation will be Dr. R.L. Goswami, Senior Principal Scientist from CSR NIST. Actually, as we were here from NIST, and we are not putting first of all from our people to present, but that list has given by Mr. Das, so I am not at all biased for this area. Thank you very much.
We are talking much on the Jorhat area, so that's why I put it like this. And I am aware that it will be a multi-sectoral brainstorming session, which will not be basically a very technical session, which where I am accustomed to being a chemist. So I try to make it so that it is. I try to, since it is a knowledge exchange session, so I try to make it such a way that everybody can give his own comments. Whatever knowledge I have, I am trying to put it here. So my topic is from inorganic adjuvants as groundwater purification media to arrest toxic anions like arsenate and fluoride. So I am trying to divide my presentation into different parts. It will be only 12 presentations, but I have a part A, part B, part C, part D, part E. First of all, I am trying to make a question whether the arsenic, whether there is an alternative of groundwater, specifically a Jorhat, in a place like Jorhat. Then, well, arsenic problem of the northeastern region. Since it is the first presentation, that's something I have to touch on also. And the CSR NIST initiative, whatever we are trying, we have tried two types of adjuvants, synthetic adjuvants and adjuvants from bio-origin, which are basically biomaterials from that is very cheaply available in the northeastern region. And cost details of the technology that whatever we have developed so far. And finally, the post-mitigation problem where we are interested on, this is the permanent immobilization of the toxic anions, and there also we will try to see the whole problem in a local context. So first my question is the first alternative in Jorhat, whether groundwater to be harnessed or groundwater not to be harnessed. This is something I try to dig in from the data that is available from the Department of Met, that is office, they have office there in Guwahati. There I have collected in a particular year, in the 2010, that was a period March, April, May. That was the total rainfall data. Why I take this March, April, May when talking about the rainfall? Why I do not take the June, July, August or September? Because June, July, August, we know everybody in Assam there is flood, there is heavy rain. So we are more concerned about this period, March, April, May. And when you compare Assam, we are still better off. If you go to Delhi on the month of May, there is totally dry. But in our Assam during Bihu, the rain starts. And now we are talking about the present context in 2010 or 2000, after post 2000 year, we are talking of global warming. So one of the problems that is in global warming is saying that the rainfall total, say in Upper Assam, we are having around 3000 millimeter rain per year. That's why our tea industry sustains. And that the total rainfall will, in one of the models they say that the total rainfall will remain same. But the peak period rainfall that will be in the July, August, that the peak will go down, rather the rainfall distribution will start from March to say November. That is one of the models. Probably that model is working well. That's why we are finding nowadays rainfall there in November and therefore rainfall starting way back in March itself. So that's why from that general perception, I took this data from the Department of Meteorology. That is available. That is there in the Guwahati airport. They have given me in March 2010 that they have got 90.1 millimeter rain in Jorhat. They had 282.6 millimeter rain. And they had 305.3 millimeter rain. Total it was 678 millimeter rain in Jorhat. So a rainfall amount is described as the depth of water reaching the ground. Typically, it is expressed in inches or in millimeters. 25 millimeter equals to 1 inch. An inch of rain is exactly that water that is 1 inch deep. That is a simple calculation that I have kept to keep the general people, whoever they are non-technical people, to understand the idea. Then 1 inch of rainfall equals to 4.7 gallons of water per square yard, or it is equivalent to 22,650 gallons of water per acre. Now, coming back, let me not go to for 678 millimeter. Let me think that in that period in our town there was a 500 millimeter rain, or it is 20 inch of rain. So in March, April, May, then we had around 453,000 gallons of rain water falling, or that is equal to 17,15,000 liters of rainfall. That was per acre, and that is because 1 gallon of water is equal to 3.785 liters of water. I assume that there is no evaporation loss, there is no percolation loss. According to the World Health Organization, per day requirement of safe water per person is 40 liters. These 17,15,000 liters per acre is sufficient for 42,875 persons for 1 day. Assuming that in our campus, in CSR and NIS campus, we have around 1,000 persons. This rainfall in 1 acre of roof from 1st March to 31st May 2010 was sufficient for 43 days. That means 1 acre, that we can understand in our Assam Kotha or Bigha system, that is equivalent to 15.18 Assam Kotha. That means what we call almost 3 bighas of land. So, water for 43 days from 90 days of rainfall over 1 acre of land was available for 1,000 people. So, it is the level of water we had. But yet we are going much for after the groundwater. We are starting I want to start the seminar with that note. So I would say that there is enough possibility in rainwater alternatives, but yet not explored completely. Probably we are not aware, or we are not aware that what we have, the raw thing that is available with us, but we are looking for the scientific literature, technical literature, what are the companies are feeding in, and we are going for immediate solutions by going for groundwater immediately. So this is something we have to probably, I feel, seriously go in. Now, coming to our institutes, the R&D, whatever we were involved in R&D initiatives and activities by CSR and Jorhat on research related to problem of arsenic and fluoride in groundwater. Let me show something about the background of the problem. This is the map of northeastern region. These dotted areas are full of arsenic already reported. The Titapur lies here. Then we have the arsenic there in Tripura. We have the arsenic there in Manipur in Thoubal area. And these are an extensive data is available in the Nerivalam, which works itself. So you will find it in the net also. Then of course here some proven arsenic contamination that was reported in 2004. At that time Titapur was not much walked out. So that's why Titapur is absent in this thing. But Titapur is now a very dangerous zone. It has been already established. Assam we had at that time Borpeta, Dhemaji, Dhuburi, Dorong and Gulaghar. In Manipur, the Thoubal district, Arunachal, these areas, Papumpare, West Kameng, then Tripura, West Tripura, and Nagaland, Mokoksang. Mokoksang is not far away from Jorhat. The rivers that like Kakodunga and Bogdo actually are flowing from the Mokoksang area of the Nagaland itself. Okay, now the comparison of what is globally available arsenic removal methods. This actually I took from net, so that's why it's not that very clear. I would like to say that what is arsenic, what are the different arsenic removal methods that they are taking. in the world for domestic and low cost use, and community which are low cost. Similarly, there are domestic with high arsenic removal. There are sectors again community with high arsenic removal, and they have said something for domestic and brackish-water, but of course it is not much of interest. It is generally for near the sea beaches. This type of brackish-water is available. And in our places like us, the alum coagulation and iron coagulation and enhanced coagulation, that these are some certain methods that have been specified by WHO papers, that these are both for domestic and community level. These are well acceptable or very well suitable, they say. Then here, of course, they have given a number of methods. They are wanting almost, we always talk about a membrane method. The membrane method, out of membrane, some people also will talk, but the WHO paper says that it is not very good for domestic and community system. So these are the commercially or technically developed methods. Okay. Then let me go. Let me go to what we have as a chemist, what we are working. Let me say something about the type of synthetic adjuvants that we have developed. This is a little bit difficult for people from community or non-technical people. Still, I have to say, because we had some work there. So this is basically one of the very common, very cheap mineral called brucite. It is nothing but magnesium hydroxide. You take few magnesium ions from its seed out, but it's not that easy. But think of it that way. We take magnesium ions out and we put there aluminium 3 plus ion. Aluminium 3 is a positively charged ion, and magnesium 2 is again 2 plus positive. There is a charge imbalance of 1 plus extra. So that gives an extra charge over the layers. So one giving extra charge over the layers, you are importing, making a charge imbalance in the system. That extra positive charge is important for us, because that will be used for adsorption of toxic anions like fluoride, toxic anions like arsenate. That is the type of hydrotalcite like LDH or MMH, whatever way we talk. That is the thing we have developed here in our institute. Then we have studied the different combinations of LDH with different metal ion combinations for arsenic adsorption, and in many cases we have found 99.99% arsenic removal from 0.1 ppm of arsenic. Then these are the three different compositions of mixed metal layer hydroxide. Some of them are made out of calcium aluminum, some of them are of course expensive zinc aluminum, some of them are magnesium aluminum. I would say this magnesium aluminum LDH is very much common thing in the market also. A very well-known German company called Bayer is marketing this type of material as a different purpose, that is called tile seed. They are marketing and they have made a lot of money with this compound magnesium aluminum hydrotalcite since 1930s. But changing the metal ion also we had some purpose, because we are ultimately interested for permanent immobilization. The cost of production per kg, it comes out to be rupees 100 on 2009 baseline data. So then we have of course also made the flow sheet of preparing this mixed metal hydroxide in a chemical engineering flow diagram. That is also has been also done, and it's also ready with us. Then of course that is our synthetic adjuvants. But synthetic adjuvants, I tell you, that is 100 rupees per kilogram. At some point of time, we found that we should go for cheaper adjuvants that is from locally available northeastern region sources. We took two things. This green Ipomia cornea, it pani kolmo, apnaloke hokoloe dekhi thoka gos. And then this is pedihas, thanor bakoli. From these two materials, we have from pedihas case we get a very highly efficient adjuvant. Of course, we had to do some chemical maneuvering over it, and we get pedihas gas, which is a very high silica content material. And from Ipomia cornea, we got carbonized Ipomia cornea, and this carbonizer was developed in our general engineering division along with our joint opera. This is the purifier column that we are in our present project with DSC. We are trying to design, and that of course it is a computer drone concept. We have still working on this. Then finally, this is a chemical engineering database where our colleague Tabiul Hussain is working. And this is this chemical engineering database we have developed for what would be amount of adsorbent, of course, for hypomia cornea carbon will be necessary, and how much will be necessary per day for a 720 liters per day capacity plant, that starting from 10 ppm chloride, if we go to 0.5 ppm chloride. And we do not distinguish between chloride and arsenate, because if it absorbs chloride, it will absorb arsenate also. So now I talked about permanent immobilization. This is something that you are very aware of. All these structures are standing here for last 100 minimum years, and these are for last 400 years. One of the things in these structures is that they are standing because they were made out of a cement which is not generally Portland cement we can purchase from power bag at a cost of 400 rupees per kilogram. These are all based on some lime silica reactions that were done even during the early periods of British Raj here. So, for technical reasons, for IPR reasons, I cannot say right now many things. But I would show some of the products, some of the results of our fluoride permanent immobilization that we have carried out during our last Ministry of Environment project. We have, this is the Perihas case. It's basically, this is the amount of after fluoride absorption. After fluoride absorption, we expose them to a particular sort of reaction called lime silica reaction, where we react with them lime, and then we make mold, and civil engineers will know about this very easily. And then we see their maximum load bearing capacity. Once the load bearing capacity is high, it's a very good indicator that there is some mineralization is taking place. And we find that when we take normal Perihas case, its mineralization is not that high. But when we modify it with certain aluminium chemical, we find it's increased, and when we use the fluoride as an adsorbent, its mineralization increases further. That means fluoride works as an active mineralizer, which is very proven in cement chemistry. We are up to this level. Further works are going on, and probably in next 6-7 months' time we will be able to give you further results. This is all what I had to show. So to you. Thank you very much for patient hearing.
Time is running fast. So I am now requesting Dr. Unni to present his next presentation.
Somebody said a few days before, as the publicity is concerned, we the people, including me, very poor as compared to other states. That's the reason. Even somebody said about the rain harvesting. Yesterday I was at the Defense Research Laboratory. I was with the director. You don't believe it, the portable safety tank. He showed me the water which comes out from the toilet. So clear. Somebody said that, the quantity, the color of the water is so clear. And he is pumping and reusing the water. It's coming from the, the base, and he now, the technology is so simple, he has given to the government. The Chief Minister visited, so they wanted to assemble one. So he made it with a bamboo. So most of the villages में आप जाइए, वहाँ तो आपको मिलेगा भी नहीं toilet. This is so simple, and the same water can be reused, and the sludge is microbially digested, and he has got a very good bedding of the flower and the gardens. No foul smell at all. I was standing very nearby. He took the water from his own hand, from the septic tank. Can you believe it? So we have so many devices. We have so many technologies. The thing is that people are reluctant to take up. People are reluctant to know. So with this simple beginning, I said that is there a permanent solution to have a water free of arsenic? That is what we have to say. Yes, I know the place where there is with a mapping, there is a sophisticated technologies are available. You can find out. It's very well, good. Can you, remove the arsenic? Can you give a free bottle of water to a farmer? poor man, he cannot afford 20 rupees to give 1 liter of water, जो arsenic नहीं है, उसमें कुछ कर सकता है, उसी के बारे में हम लोग को सोचना है. Okay, some of the mechanism. थोड़ा बहुत हम information में बोल देंगे जो plants होता है, because my studies are basically on the plants that is available here. Little bit of goswami said. Another one is a fish. It is also used as a bioindicator. Now this information I can share. Some of the information जो publish हुआ है, and 2 of my students are even now working with PhD. So many information you don't get now because we will see अगर arsenic remove होता है, कैसे remove होता है, कैसे detoxify करता है, किस stage में store करता है, वो भी इसमें information है. Now this is a little bit where it affects the arsenic when it goes to the body. That is what Professor mentioned. It's very important to see when the water is taken, is containing arsenic, where it affects. Some plants have been reported to tolerate high level of arsenic from water and soil bioaccumulation. In India and Bangladesh, there are different levels of arsenic as far as the WHO level. That is also there. Now this is the effect of arsenic poisoning. This was a part of a project, a very small part we took it. It's a network project where it affects where. Now this is not done by me, this is available in the internet for your kind of information. For you nowadays, many people give lecture just copying or cut and paste from the internet. So this is also I have done it just to see as far as I know I expected, even I also talked to Goswami 2 days before. I copied to that, bring some doctors, and it is a very good thing that we have a doctors here. Now we have also done the epidemiological studies as a part of the project. We go with the health survey. Those areas the problems are having. We used to interact with them with the common language. The questionnaire, this is as per the epidemiological because this is had to be approved by the institutional ethics committee. Assamese में हिंदी and English वो distribute करता है, उन लोगों को problem हम लोग study करता है. क्या है इन लोगों का problem? Water use करता है, fruit use करता है, food use उसमें all are arsenic are there in addition to the other because this is a part of a project. Many other things have to be involved and based on the health survey we go with medical card. तो ये भी एक प्रोजेक्ट का जो सोसाइटल है, that is what the good thing of this laboratory serving for the 8th stage. We do as a part of the, the network projects and the societal benefit to the farmers. Even some of the interior places, they have seen, they are going to have the health camp for the first time. हम लोग उनको ट्रीटमेंट एडवाइस करता है, फ्री मेडिसिन देता है. So this is keep on going in. So we have to go very systematically in the studies. That is very important. Once you do the very systematic studies with the help of doctors, clinical doctors are needed. You have to have a very great survey what is the impact of that by using this. Then, let's see that.
Excuse me.
Yes.
Where was the study done?
I did. This is some of the areas where oil drilling sites and then coal mine sites are also there and in pepper. Now you can see here some of the parameters. This is from the sample collected from the oil drilling sites. You can see the red color one, high concentration as compared to the WHO is given below. But these are the area. I'm not going for the specific for cadmium or what they may be there, but we are concentrated what are the parameters we got. But again, it's an ongoing work. Now, this is the mineral analysis of water. You can see the arsenic concentration here, oil drilling site, as compared to the WHO, about 4 times more. So now, how we got this information? Based on the parameters, based on the interaction with the people, where they are drinking the water, what sort of a water they are using it. They are collecting from शायद you call it टुकड़ी जो बोलता है वहाँ से जो पानी लेता है या ट्यूबवेल से लेता है, तो वो सब हम लोग कलेक्ट कर लेता है. उसके बाद मैंगनीज कितना है, उसमें भी हायर कंसेंट्रेशन मिल रहा है, but I am not interested on that. Then उसके बाद जिंक है, वो भी ऑलमोस्ट ठीक है, but आर्सेनिक कंसेंट्रेशन में बहुत ज्यादा है. ये हम लोग को मिला तो हम लोग को ये सोचना पड़ा इसको किस तरह से रिमूव कर सकता है. There are big filters are coming, but what are you doing with the filter? There are community based filters are being used. आपको पता है जो जगह पर 5 ppm आर्सेनेट है, maybe 2 ppm. There are places 10 ppm. When you go for a big filter, entire arsenic, the water with the contaminated arsenate is pumped to the filter. You get a potable water. But The filter is contaminated with the very high level of arsenic. Where you will dispose it? You will dispose in the space? No. You will dispose, maybe go in the night, maybe after 2 years, maybe in the Brahmaputra. Again, I can tell you a reason. This is the social aspects what we have to consider. You know, the murtis are prepared. Very beautiful murtis. You know the concentration of toxic metals there? Mercury and arsenic? As a religious point of view, yes, we worship. After the worship, what we will do? So it's a personal level, we can make a lot of control. You put it in the water, arsenic is again coming. It's not like the groundwater. So this is what I was telling you. Filter, it's very good for a short-term basis. अभी हम लोग को ये सोचना है long-term basis पर. As on today, there is no device. I can challenge for this. If anybody can come up, I can remove it with a bio, eco-friendly, non-toxic substances which can 100% remove the arsenic. It is not there. Why? You go to the net. Why it is so? Research is going on. A lot of monies are being pumped. It is very good thing that, at least just a couple of days back I got information that there is an NGO working on it. That's the reason I took a special interest to talk, though I'm not a man of chemistry, I have a chemistry in BSc level only. So this is what I wanted to share you. Then we took the plant material. Now everybody, it's simply presenting a data does not work. This is again an hypobia. You can see the control plant and you can see the different levels of ppm level of arsenic used. And you can see 7 ppm for 15 days. Some sort of this experiment is still going on. There is no problem, as that means this plant has got some sort of a capacity to take this arsenic and then retain and detoxify. Like in the beginning I said, water hygiene can take the mercury. We have done an experiment because one of the enzymes here also, we are looking on this aspect, can detoxify where this arsenic is formed as a complex and a non-toxic form which acts as a cofactor. Now what happened? I can tell you a little bit. To those who are science level, when you have an enzyme is a molecule, one part, we call it the active sites. What will happen? All these enzymes repair metals. वो एक cofactor में उसमें complex बनता है. Then only the enzyme will be active. Now this arsenic is arsenic in the form of a triol complex. I'll let you know. This is in the very preliminary stage of studies. We have to do further in the enzymatic pathways. Now, here you can say again with the 3 ppm, there is no problem. It can go very well. 7 ppm, there is no problem. 10 ppm, up to 10 ppm, you can see that coming to the 15 ppm, you can say after 1 month of treatment, continuously in the tank. See the 15 ppm. The leaves are gone. See, now again with a very higher concentration. Now what will happen? Bioaccumulation. What will happen? It takes from the roots, groundwater, whatever it may be, again detoxified. Up to some extent root is used, converted. Then it goes up along with the xylem or phloem. ये plants में होता है, उसमें जो कोई भी न्यूट्रिएंट, आप सोडियम हो, पोटासियम हो, कोई भी बायो फर्टिलाइजर डाल देता है, पत्ता आपका बहुत हरा बन जाता है. कैसे हरा बन जाता है? वो ये जो न्यूट्रिएंट है, ये खींच लेता है. इसी तरह arsenic को भी खींच लेता है. खींचने के बाद ऊपर चले जाता है, एक कॉम्प्लेक्स बन जाता है. Then you can see it here in the cytosol. एक plants में parts होता है, वहाँ पर वो accumulate हो जाता है in the physical storage. और ये plants उसको utilize करेंगे and it will start growing it. So up to some concentration of arsenic can be accumulated as a part of it, you can say the wild, as a medicine to the plants, and plants will keep on growing it. Arsenic is removed. Now, the previous slide, this will also a very good indicator for a farmer. Suppose the farmer, उसका कोई knowledge नहीं है और ponds में arsenic hit नहीं है. Suppose the society like us, किसी ने आपको बोला कि देखिए भाई आपका पानी हमने एनालिसिस किया, उसमें arsenic है. अब उसको कैसे पता? पानी देखने में तो सेम है. This is plenty available in this part. You allow them to grow. इसमें तो कोई साइंस का जरूरी नहीं है उसको, वो प्लांट्स को दे दीजिए. देखिए उसमें कितना लेवल तक ये ग्रो हो जाता है. अगर वो ग्रो होता है, इसका मतलब एक तो इंडिकेटर हो गया, इसमें आर्सेनिक है. फिर भी अगर आर्सेनिक ज्यादा है, immediately that what you can see it here. See here, if it is more than 15, see the condition within 3 days and 25 ppm. Now you can see it 50 ppm, very high concentration, completely within a day, in hours interval. किसी ने बोला कि आपको कुकरी और पानी है उसमें पूरा आर्सेनिक है. अगर ग्राउंड वाटर पानी कलेक्ट किया उसमें पूरा आर्सेनेट का रिकॉर्ड इंफॉर्मेशन है, you allow the plants to grow. See in the condition of 100 ppm and about 50 ppm like that, it completely goes. So this is, we call them as a bioindicator. Now what will happen? What does the plant system? Plants क्या करता है ये भी जरूरी है. As a scientist point of view, as a farmer's point of view, they never bother. कोई भी चीज में उन लोगों को ये नहीं चाहना है कि इसमें क्या है, उन लोगों को चाहिए प्रोडक्ट. सपोज सिल्क है, दे वांटेड टू गेट ए बेस्ट सिल्क. ये नहीं है कि उसमें प्रोटीन है, प्रोटीन बोले से उन लोगों को कुछ पता है. तो हम लोग को ऐसा डिवाइस निकालना है, वेरी सिंपल, दैट कैन बी इजीली ट्रांसमिटेड. बहुत इजीली, जो आपका जो गांव पंचायत है, बुरा कोई भी हो, उन लोगों को इंफॉर्मेशन दे सकता है. इसमें क्या होता है, ए पोर्शन ऑफ द आर्सेनेट टेकन अप इन द प्लांट्स मे बी हेल्ड इन द सेल वॉल्स. दिस ऑल हाइपोथेटिकल. इसको अभी प्रूव भी करना. अभी हमने थोड़ा प्रूव किया प्लांट्स की एक्सेस रूट के ऊपर. देन द रिमाइंडर क्रॉस रूट मेम्ब्रेन्स प्रोबेबली बाय ट्रांसपोर्ट. दिस इज व्हाट हैपनिंग. जो केमिकल जो फिल्टर होता है उसमें भी यही होता है. देन फैसिलिटेटिंग द अपटेक ऑफ फॉस्फेट आल्सो हेल्प. आर्सेनेट को प्लांट्स में रोल भी है. हमारा आपका बॉडी में भी आर्सेनेट है, लेकिन अप टू सर्टेन लेवल. व्हेन इट गो बियॉन्ड दैट, देन ओनली आपका फूड पॉइजनिंग होता है. इनसाइड द रूट सेल, some arsenate is reduced to arsenite, a small fraction of arsenate, and as they move from root to leaf via xylem, जो हमने आपको बताया. उसके बाद reduce the enzyme by the enzyme and then forms a complex. जो a complex बन जाता है, that is becoming non-toxic. वो vacuole है plants में, छोटा छोटा होल टाइप का है. ये तो आपको दिखाई नहीं पड़ेगा, but as a farmer's point of view, you are not supposed to know that. Plants को तो grow होगा. But this is happening up to some level of concentration only, not beyond that. But this information I'm giving to, we are also working on that level. As a research lab, you can also look into that. Some of the studies what I said, आपका जो पुकरी है, अगर किसी ने बोली उसमें आर्सेनेट है, you take this plant called, I don't know, I think I was told just now that it is also called hypomia in Assamese, like जो terminal chebula, you call it a helica. But here I was searching so that जो farmers को पता चले ये plants को आप कोशिश कर सकता है. जो कहीं पर भी arsenic contaminated water है, you take it and check it out. So this is the mechanism. Now coming, what will happen at a molecular level? As a molecular, nowadays everybody talk about molecular biology and genetics, biotechnology. ये कुछ होता है कि नहीं है, molecular level पर क्यों होता है? Same thing happening in human being. What do you mean by toxicity? What do you mean by the poisoning? This is what happened. You can see 15 ppm, 1 ppm, 100. जो higher content जो DNA होता है आपका genes, सिंपल लैंग्वेज में बोल सकता है, जो आपका जो बॉडी में जीन्स जो होता है, डीएनए होता है, वो डिग्रेडेशन होता है. दैट आल्सो बी स्टडीड. अभी देखिए 1 ppm में कोई डिफरेंस नहीं. इसका मतलब 1 ppm तक प्लांट्स लेने से उसके कोई चेंज नहीं होता है, वो ग्रोथ होता है, इसलिए आपको दिखाया वहां पर. लेकिन 100 से होता है, इवन 15 से शुरू हो गया. दैट मीन्स द प्लांट इज गोइंग टू बी एवरी सिस्टम. It is happening, whether it's the plants or the human body, it is being degraded. So here it is a degradation. This is happening in the plants with a certain level of, at some molecular level. Now we have taken up another very interesting thing you must be know. I don't know in Assam what you call it. Chena puncture. This is also a fish. One of my research students is working at the Unni University as a PhD problem. We did it that this fish is quite common and I think it is a little bit costly. We did some work up to 8 ppm. To 10 ppm, it can survive. Now, somebody may ask, person like doctor is sitting here, whether did you taste this thing or not? No. We have not tasted with the fish, but we have seen the survival of this fish. Now, at different concern, this is also we call as a bioindicator. Suppose the fish you brought from an arsenic, or you have a fresh fish. Somebody said that, pani bahut zyada arsenic. You allow the fish to grow, what will happen? You can see it here. See the different color. This is the control fish. Now the fish exposed to the arsenic about 1 ppm, you don't find any changes. So the pots में जो वो fish को निकाल दीजिए, तो उसको पता indirectly आपको पता चल सकता है कि up to this level, see the decoloration, and you can see 50 ppm and the same fish. Fishes were ponds exposed with arsenic up to 10 ppm survived more than 1 month, and above concentration you can say that within 24 hours. Now, a very detailed studies are here. We are looking for it. This is also, we call them as a bioindicator, as a farmer's point of view. They cannot go for afford to a filter. They cannot purchase it. But they can give a message. It contains arsenic. It is toxic. But now, this fish can take up to 10 ppm. We have studied about 1 month. What will happen? What will be the detoxification? How the fish is survive? If the fish can survive, if the same thing human body is consumed, human is consumed, what will happen? We have to study. Now, we also studied at the molecular level. So the fish, when it's taken, you can see the DNA. Here, this is 100 ppm. Entire DNA has been degraded. Those fish which is not survived, dead within 24 hours. That means at a very high concentration, you can see 30 days exposed. It's completely degraded. The same thing happening in human body also. That we indirectly, the doctors' words, they call it food poisoning. And in the serious, there is a mortality also. So, but at the low level, nothing, fish is surviving. You don't find at the genetic level also. Now, what are the enzyme parameters, where it works, how it works, whether the fish can be consumed by the human being, anything is happened, that detailed studies are needed. But my point is that just to use a bioindicator and then the fish can take up. We have studied the fish or the plant allowed to grow in water, उसके बाद plants को निकाला, वाटर में जो कितना आर्सेनिक हम लोग स्टडी किया उसमें बहुत पूरा they have taken up, consumed. So that is the one thing what I wanted to share. Now, this is the other parameters. What will happen with the controlled and 5 ppm? Entire metabolism. जब खाना खाता है, जो चावल में कार्बोहाइड्रेट है, प्रोटीन है, लिपिड है, जो कोई भी चीज हो जो आर्सेनेट पानी से भी, फिश में भी वो कंटामिनेशन होता है, इसीलिए फिश मर जाता है. पूरा एंजाइम्स में उसमें हायर कंसंट्रेशन में. So whether it is a human being or in fish or anything else, at a higher concentration it is toxic. But detailed studies are needed. Now this is it. You can see with 1 ppm, 10, 25, 50, 100. So up to 1 to 10 ppm, there is not much changes at the genetic level. But if the fish is consumed by the human being, you have to see that. A very systematic study. Here also the metabolic pathways has to be studied. So this is what I said. What will happen? I immediately jumped up to the molecular level because the genes are very important as well as human being or animal system. Anything is concerned, whether it affect or not. But again, I have to say that this will be a bioindicator. Bioindicator का मतलब वो fish आप पानी में डालने से आपको indirectly पता चलेगा अगर fish survive होता है इसमें ज्यादा नहीं. जो किसी ने आपको बोल दिया कि आपका water में आपको test भी करना है side by side. Fish को biological system है, but you have to see chemical because that will give you an accurate result very soon. Now I will show you one devices. Now what will happen? Plants were found to survive more than 2. This is almost the conclusion of that up to 15 ppm. A decrease in chlorophyll content, ये जो studies है, जो पत्ता पीला हो जाता है, ammonia can be used as a biomarker, I said. मतलब वो plants को allow करने से up to 10 ppm तक, अगर 10 का जगह 25 है, within 24 even 2 to 3 days the plants will die, वो मर जाएगा. That biochemicals in fish में क्या होता है? To survive more than 16 days up to 10 ppm. Now fish will survive, but you do not know if the fish is consumed. You have to have a very systematic studies that comes under a part of a clinical part. You have to go with a lot of family. Who will go to take up the fish which containing 10 ppm? It's very difficult. So we have to study very systematically where this arsenic is being accumulated. तो उसमें आपको study कर सकता है. Degradation total, all the parameters जो higher concentration में है, उसमें कम हो जाता है. Then DNA fragment, that is very important. जो concentration ज़्यादा होता है, आपका body को change होता है, उसी तरह fish में भी होता है. Now, the detection mechanism. Conclusion. Significant finding from the study sites. Numerous studies confirm that toxic metal have damaging effect on all the cells of the body and directly impair neurological, immune, endocrine, digestive, respiratory and detoxification mechanism. Heavy metal poisonous, disrupting our cellular enzymes, जो higher concentration में होता है. But our future plan of studies, that what again I am stressing, we are doing it. This is you can say about 0.1% contribution as a bioindicator जो हमने आपको बताया. Must be directed. Any sort of discussion you are going to have it here should focus how to eradicate, how to remove the arsenic permanently and how to detoxify without dumping this material in the other part. To find out a permanent solution to remove arsenic-based bio devices on long-term basis, not a short-term basis. So that is what we have to look into for. This is a pass to the strength. Thank you.